Children, in this chapter, the important concepts are meaning and discovery of cell, cell theory, parts of cell. In this capsule, we will know about meaning and discovery of cell, microscope, under that simple microscope and compound microscope. Cell theory, under this process of osmosis. Children, let us start with an activity. Classify the following into unicellular and multicellular organisms. Will you classify now? Have you done with the answers? Good. Now we will check it. We all know that there are varieties of living organisms around us. All living organisms are composed of different parts. Children, now can you name the parts of the plant? Yes, you are right. Stem, leaves, bud, branches, flowers. Yes, you are right. Fruits, seeds. I hope you have named all these parts. You might have named other parts also, isn't it? Good. Now, can you name the body parts of animals? Neck, legs, horns, etc. All the parts may be different from each other, yet they are composed of tiny units called cells. Children, let us understand more about cells. Cell is the smallest part of all our organs, which is capable of independent existence and performing the essential function of life. Can you define a cell now? Yes, cell is the fundamental, structural and functional unit of living organisms. Now note this definition in your notebook. All living organisms, including ourselves, start life as a single cell called the egg. If a cell is the building block of all the organisms, how come we never noticed them before? Yes, that is because you can't see a cell with your naked eyes. The important characteristics of a cell are: you are right, cell is capable of performing all vital life activities. Cell cannot be seen with the naked eye. Yes, you copy these points. Therefore, it was necessary to invent an instrument to magnify and study the cell. Children, this led to the invention of the microscope. You know the microscope? The microscope magnifies the object so that can we see the tiny ones clearly. Children, Antony von Leeuwenhoek is said to have constructed 400 microscopes. Some of these microscopes had a considerable magnifying power up to 200 times. All his microscopes consisted of a single biconvex lens and were called simple microscopes. Yes, copy this. Later, Robert Hooke, an English scientist, used two lenses for better magnification. Such microscopes were later known as compound microscope. Copy this. Let us understand the experimental setup of his experiment. Robert Hooke, in his experiment, placed the object to be seen on the stage below, and light from an oil flame was thrown on it by means of a concave mirror. Now, let us watch the video of Robert Hooke's experiment. Robert Hooke discovered cells by examining slices of cork from the bark of a tree and observing it under the magnifying glass. He was able to see partitions boxes. He called them as cells. The word cell means small room. Modern compound microscopes look like this. This ordinary compound microscope is greatly improved design of the ordinary Hooke's microscope. It has an eyepiece lens through which we see the object. It has an objective lens which is facing the object. It has a mechanical stage on which the object is mounted. The electron microscope added future to the unknown facts about cells. How is electron microscope different from compound microscope? Yes, electron microscope can magnify to over 2 lakh times as against the ordinary compound microscope, which magnifies an object up to maximum of about 2000 times. Compound microscope uses light, which is bent by glass lenses to magnify the object, whereas electron microscope uses beams of electrons which are bent by magnets. Now it is the time to think and answer. Yes, here is a question. Name the following. First one, the scientist who invented the first microscope. Second question, the kind of microscope that consists of single biconvex lens. Third question, the kind of mirror used for throwing light on the object in Hooke's microscope. Yes, you are ready with your answers. Shall we verify the answer now? Answer. The scientist who invented the first microscope is Antony von Leeuwenhoek. For the second question, the kind of microscope that consists of a single biconvex lens is simple microscope. Yes, for the third question, the answer is mirror used for throwing light on the object in Hooke's microscope is concave mirror. Children, here is one more question to think and answer. What is the maximum magnification that can usually be achieved by first one, a compound microscope, second one, an electron microscope? Yes, what is the answer? You are right, a compound microscope magnifies 2000 times, and whereas an electron microscope magnifies 2 lakh times. Children, in the year 1839, Malthias Sheldine, a botanist, and Theodon Schwann, a zoologist, proposed the cell theory. According to cell theory, there are 3 major points. All living things are made up of cells. The cell is the smallest living thing that can perform all the functions of life. Children, all cells arise only from pre-existing cells. Yes. Now you copy all these points. What does the cell theory mean? Yes, if you observe any parts of the plant or any part of animal under microscope, it will show cellular structure. All the activities, any in them, are due to the activity in the cell. Cells continuously die and are replaced by new ones, which are formed by division of younger cells, which means cells are formed from pre-existing cells. Yes, copy these points. The life of both plant and animal started as an egg and seed, respectively, which are nothing but cells. Children, think, why are cells smaller in size? Yes, cells are generally smaller in size for two main reasons. That is, different regions of a cell can communicate with each other rapidly for the cell to function effectively. Cells have a large surface area or volume ratio for greater diffusion of substances in and out of the cell. So this is a very important point. Copy this in your notebook. Now let us understand how molecules move in and out of the cell through an experiment. Let us take a jar of water. Add 3 to 4 drops of green dye into the water. Children, now observe the change in the color of water. Whether the change was gradually or immediate. You see that the change is gradual. This is due to the movement of molecules. Green dye is getting diffused. The process of movement of molecules is called diffusion. In diffusion, the molecules move from a region of higher concentration to a region of lower concentration. Now let us verify whether molecules move from higher concentration to lower concentration. For this, children, observe this experiment carefully.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Can you define osmosis now? Yes, you are right. It is movement of water molecules from the region of higher concentration to the region of lower concentration through the semi-permeable membrane. This kind of process is called passive transport. So this is once again an important definition. Note it down in your notebook. Children, sometimes the molecules move from lower concentration to higher concentration also. This process requires energy. This is called active transport. You have understood the meaning of cell, discovery of cell, and about cell theory. You also learned about the process of osmosis in this camp. Children, here is an extended activity for you. Will you do this activity? Take a perfume bottle, open and keep it in one corner of a room. Observe the change in the room